In this video, we are going to talk about the synaptic transmission. First of all, a few terminologies. I would like to draw a diagram to mention the basic terminologies about the synaptic transmission. So this is a neuron. That's an axon of that neuron. Axonic terminals. And the nucleus. The dendrites, the myelin sheath, so we know that in the nerve transmission the neurons are linked with each other in a way that there is a gap in between. So the second neuron in the synaptic transmission involved similarly the cell bodies, dendrites, so in this transmission this neuron will be known as presynaptic neuron and this one is post synaptic neuron, and this gap or this place between the two neurons is called a synapse, and the gap in between the presynaptic neuron axons and postsynaptic dendrites is called synaptic cleft. And all the communication between these two neurons will happen chemically, and these chemicals are known as neurotransmitters. Like acetylcholine is the most commonly known neurotransmitter. Okay, so now let's talk about how this synaptic transmission happens and how does the action potential, which has started from the cell body and axon hillock of the one neuron, has to be shifted to the next neuron through these synaptic clefts. So let's see it in the details. If I zoom into this part of the neuron, and this part, right? So this is the axonic terminals and this is the cell body somewhere. The mitochondria provides energy to produce the specific protein-like enzyme-like structures that are called neurotransmitters, and those neurotransmitters are packed into the small vesicles. These vesicles, once filled and packed with the neurotransmitters, start moving towards the end into the axonic terminals. Once they reach to the axonic terminals, they wait there until the action potential initiates from the axon hillock and moves along the membrane to the end, moves towards the end, and then they open some specific gates. So this specific gate is permeable to calcium ions only, and hence they are known as voltage gated calcium channel. Once these channels open, there are several calcium ions present outside in the synaptic region and synaptic cleft. They rush into the axonic terminals and then they get attached to specific receptors, allowing the opening of they attach to the specific receptors and allowing the diffusion of the membranes of the vesicles with the neuron membrane, which results into the release of the neurotransmitters in the synaptic cleft. Whereas the postsynaptic dendrite, this one, they have specific channels for sodium and potassium. They are known as ligand gated sodium channel and ligand gated potassium channel. These neurotransmitters comes and attach to these specific receptors, resulting into opening of these gates. The sodium potassium gates open when neurotransmitters attaches to the ligand gated sodium and potassium channels. So once they open, they allow the movement of sodium ions from the outside into the dendritic and the somatic regions of the neuron, and potassium ions from inside, they start rushing out, hence reversing the membrane potential, just as we have discussed in the generation of the action potential in the previous video. So once this type of the potential is known as graded potential, so they result into the generation of the graded potential. So the graded potential moves towards the cell body of postsynaptic neuron to the hillock, and if the graded potential is greater than threshold potential, it will be equal to action potential generation, right? And then the rest mechanism is the same as the flow of the action potential. The way it has moved from this neuron to the end, the same action potential will start moving towards the end of the next postsynaptic neuron. Once that action potential is generated, the postsynaptic dendrite releases the retrograde proteins that go and attach to the calcium channel and resulting into the closure of calcium channels, so that the diffusion of the more calcium ion is stopped and ultimately the release of the more neurotransmitter is stopped. This is the negative feedback mechanism. And then all that neurotransmitter which is left unused in the synaptic cleft, so they are picked by specific proteins which are known as the transporter proteins, and they take them back into the cell, and that this protein is either moved back, all these neurotransmitters are either moved back towards the cell body for recycling and repacking into the new vesicles, or they are themselves again packed in the new vesicles and are released again when the new action potential arrives at these axonic terminals.